the models that we are talking about in this workshop, the purpose is to incorporate biological complexity systematically and to look at multiple applications, like we were discussing earlier, not just a single, but multiple applications, multiple metrics. And so I'll tell you today about what we have done in that front at the Allen Institute, building our models of the primary visual cortex, area V1 of the mouse. And we hope that this is a part of a growing trend in the community, and that more and more people will use these models and other models that are becoming available, and that moves the field forward. So there are 2 points, I would say, two major points about multipurpose models. And first is integrating various multimodal data about the brain, or in this particular case, about the cortical circuit into this model. And the second part is applying the model to multiple questions, right? And so let's talk about these 2 aspects and start with the first one, integrating the multimodal data. So this is what we have done. Like Gautu said, we published our paper earlier this year. It's been a work of a lot of people, actually. So it was spearheaded by the scientist in my team, Yazan Beleh, and then other people in my team also participated, such as Kael Dai, Nathan Gowans, Sergei Grati. It was also a collaboration with the team of Stefan Michalas, also at the Allen Institute, with contributions from Ram Iyer and Bin-Huan Cai, and several other people, and many experimentalists have contributed. So this really has been a big effort. The result is shown here. So we are focusing on the central portion of mouse V1 here. The choice of V1 is because that's where most of the data are available, at least for the Allen Institute. That has been really a focus of our research over the last several years, and a lot of data have been accumulated. So the models that we published are illustrated here. They contain over 230,000 cells. They span all the layers of the cortex from layer 1 down to layer 6. And they contain a lot of complexity in terms of the biological cell types present here. So it's 17 different cell classes that we model explicitly in this model. And there are 2 versions of this model. So there is the biophysically detailed one and the point neuron one, and there is a one-to-one mapping between them, so that there is a counterpart for every single neuron and every single connection between the model on the left and the model on the right. And for the biophysically detailed version, of course, it requires hundreds of CPUs to run a simulation, and maybe you can get about 1 simulated second within 1 hour. But for point neuron version, it's actually accessible to a lot of labs that may not have a lot of computing resources, because you can run such a model on a single CPU and get 1 simulated second in just a few minutes. And finally, these models are freely available. We've made them publicly available before publication, and we really encourage the community to use them for their purposes, for different, try to use them for different questions, and of course, give us feedback. We want to improve them and make them better in the future. So in terms of building these models, there were several steps, and one is the building blocks. And I must say here, for the purpose of building such models, I'm really fortunate and privileged to be a part of the Allen Institute, where a lot of this very interesting data are being obtained. On the other hand, of course, the Allen Institute shares all this data freely, so anyone can, in principle, do that. So we start from this database of the cells of neurons, for which we have electrophysiological and morphological properties recorded from individual neurons. Actually, now also there is a data set that contains also transcriptomic characterization of those same neurons. We developed approaches to build models of such individual neurons at different levels of resolutions, and that provides a library of these building blocks of individual cell models which we then sample from to fill in the model with realistic neuron density, all 230,000 of them. The next step is connectivity. And again, if we think about the data that are available, there are really cool data sets that have recently been made public from the Allen Institute. There is the data set on synaptic physiology, so a characterization of the connectivity between different cell types in V1 using multi-patch synaptic physiology measurements. And then a data set on electron microscopic reconstruction of currently a portion of V1, and soon a whole V1 will come. And you will hear about this amazing, really systematic and very powerful data sets later in this workshop. And of course, the idea is that we take the data from these data sets and put them together into characterization of the connectivity in our model. However, when we were constructing these models about 3 years ago, those data sets haven't been ready yet. And so instead, what we did in the current generation of the model is to do a really thorough survey of the literature. And basically, we went through all the papers that we could find about connectivity and synaptic strength and axonal delays and synaptic targeting of dendrites, especially in the mouse V1 and other cortical areas, but sometimes we had to go to other species like rat and put together what is perhaps our best guess at the connectivity in this part of the cortex, given the state of the literature, right? So we put that together. By the way, we make this metadata also publicly available together with our model, and that's how the connectivity is built in. Then another part of the model is that we need to provide inputs. And this being the primary visual cortex, the main input is a visual. So this is the pathway from the retina to the lateral geniculate nucleus, or LGN, of the thalamus, and then to V1. Because there is not enough data to model this explicitly, we model that as a layer of filters. So filters that operate in the visual space. They can take in any visual stimulus, any movie, as an input, and convert that into a train of spikes, right? And then there is a lot of these filters of different types distributed in the visual space, and they project to individual V1 neurons. And the way how these filters are parameterized and how they project to V1 neurons has been really carefully parameterized using data from in-house data and from the literature. So again, a very realistic model to the extent possible. And the good thing about it is that with these filters, we can use arbitrary visual stimuli. So we can use the exact same stimuli that are being used by our colleagues, experimentalists, when they run their experiments, so that we can compare apples to apples between the model and the simulation. And so that brings me to the second part, which is applications, right? And multi-purpose applications of these models. So let me walk you through a few examples. First, what we did in this actual paper that was published already. Again, here we were leveraging an amazing data set that you will hear soon about also in this workshop of neuropixels electrophysiology recordings. So these are novel probes, very powerful, that provide a lot of throughput from experiments. And give you a lot of information, for example, about spiking activities of neurons in V1 and other parts of the brain. And so our colleagues obtained these recordings under a variety of visual stimuli from mice that are awake, that watch something on a computer screen. And then, like I said, we can use exactly the same stimuli and run similar simulations. And then we need to compare what is obtained in the experiment with what is obtained in a simulation in this spike raster example. And here we are looking at just the central part of the model with 50,000 neurons spread across all the layers. So the key here is that we are talking about multi-purpose models. And so we want to look at multiple metrics for multiple stimuli. And here how it goes. So this is an example of responses to drifting gratings, and we can look at a few different things, for example, at the firing rates of individual neurons or at their direction selectivity. And what we can do is to build distributions for neurons of different populations, right? And so here we look at layer 1, 2, 3, and so on, with different populations, excitatory or fast spike and other inhibitory, and compare between simulation and experiment. So this is the distribution and the experiment in gray. Blue is the biophysical model, and orange is the point neuron model. And well, this is like a graphical representation of these distributions, but what we actually do is we develop a score for each population. For each metric, we can develop a quantitative score telling us how different those distributions are. So I'm not going too much into details, but here you can see that the simulation and the experiment are in agreement more or less. It's not ideal, but it's pretty close. And also the blue and the orange, the point neuron and biophysical models also are pretty close to each other. And then we can look at other stimuli. I mean, again, this is all described in the paper, but just very briefly, we can look at full-field flashes, for example, or a totally different class of stimuli, which is natural movies, right? And we can look at other types of metrics, such as signal correlations, for example, or time to peak in response to the flash, or correlation of signal and noise correlation, or lifetime sparsity. And you see that for some of them, and for certain cell populations, we do better, and for others, maybe we do worse. Here we can see that, for example, the model is quite a bit slower than the experiment in response to flashes. And so this is good. You know, this tells you that some things are captured well in the model and others require attention, and that's something probably where we should really focus on for the next generations of the models. Also, the work like this allows you to do predictions, make predictions using models. And so again, we describe several predictions in this paper. All of them have to do with the logics of connectivity, the functional properties of connectivity in the circuit. So things like like-to-like connectivity, where neurons that prefer similar stimuli have a stronger and more probable connections between each other than the neurons that prefer orthogonal stimuli. And that's something that has been described experimentally for certain types of neurons. But here in the model, because we didn't have this information for everything, we have to make a lot of assumptions about how those rules work for different cell types. And eventually we found some variations of those rules, which gave us good results in terms of, for example, direction selectivity. And so then that constitutes a prediction from our model. So we see, for example, that these rules are particularly important for SST and VIP neuron populations. We find a variation of this rule that worked really well in our hands, where for the excitatory cells, let's say we have this target neuron here, and it prefers this direction of movement to the right. And then in this rule, the excitatory connections to this neuron tend to come from a stripe that's perpendicular to this preferred direction. So that really helped to make our model work well. And then at the same time, this paper appeared on bioRxiv from the Karandini and Harris lab, where they showed basically the same architecture experimentally using rabies experiments. So that was very encouraging. So you can make predictions like that about structure-function relationships. And then you can go further. And now we have a number of collaborations using this model and some follow-up work. And I think there much more can be done. I'll show you a couple of examples. So this is now, again, a continuation of our internal collaboration with the team of Stefan Michalas and spearheaded by Bin-Huan Cai. Perturbations, right? So perturbations are a really important tool. And in the model, you can have a lot of control of what kind of perturbations you do. So here's an example where we have an intact simulation, and then here a perturbed simulation where we suppress activity of all excitatory cells in the layer 4. And you see that has really strong consequences on the firing, right, from this intact simulation to the perturbed simulation, a huge perturbation. Well, we can do a lot of different perturbations, and later today you will hear from Chris Harvey. I think he will be talking actually about something else, but he published a really cool paper in Nature recently, where experimentally they showed a perturbation of single neurons using optogenetics, right? And so what they found was that when you perturb a single neuron, there is such a sort of center surround effect where neurons close to that neuron are excited if you excite the layer 2-3 single neuron. But the neurons that are further away are suppressed. And we observed the same thing in the simulation, which is very nice, you know, very, very similar shape of the curve when we test that type of perturbation. But now, of course, we can go further. And here is an example from this paper that we have on bioRxiv, where we perturb multiple neurons in layer 2-3 of our model under different conditions. So here we go from 1 to 100 neurons, and those are neurons that are all in approximately the same place and that all prefer approximately the same stimulus. And then this orange and blue curves, they describe what happens to the other neurons in that area, in that place, that prefer similar stimuli, right? And what we see is that under condition of a strong input, high contrast, the activity of those neurons is suppressed when we perturb these other neurons. So what this suggests is that we are observing something like efficient coding here, where you know, the other neurons are already active, they tell something about the stimulus, and then there is no need for yet more neurons to be excited here. On the other hand, if we have a small contrast, such as 5% here, so different type of stimulus, so weak stimulus, that effect is actually opposite. So activity of those nearby neurons, similarly tuned neurons, goes up. So this is now more like a robust coding situation where it's actually beneficial to add more voices to the crowd. So this is interesting. Obviously, this is a prediction from the model that yet needs to be tested experimentally. But it's an interesting prediction suggesting that the model can operate, and probably the cortical circuit can operate at least in 2 states. Under some conditions, it's more like robust coding. Under other conditions, it's more like efficient coding. So one can use these models to try to make predictions along these lines. And then another example is something that Gaute mentioned, our collaboration with his team, spearheaded by Atle Rimehaug, and his participation from Alex Stasik and Jordan Ness. So it's not just the spikes that we can extract from these models. It's also signals like the electrical fields, for example, right? So we can compute electrical fields. So we can take the low frequency component of it, so the local field potential. We can convert it to the current source density. So do exactly the type of things that are done experimentally from those same neural pixels recordings, computing the electric fields. And then we can compare, again, apples to apples, local field potential or CSD from experiment with those from the simulations. At the same time, we can look at the firing rates in different populations and different layers. Again, an experiment in the model. And this all shown here is in response to a full field flash stimulus. So what we find is that the model that we published actually didn't do such a good job reproducing this type of metrics. It wasn't horrible, but we found ways to improve it. And so this is sort of the agreement that we have now. This is still not ideal, but you see that there are certain features here that are similar between the model and the experiment. And we have a whole set of quantitative metrics which show how different the model and the experiment really is. And in doing so, we can do specific changes to this model to improve this comparison. And so this again constitutes prediction of you know, what it is that needs to be present in the circuit to reproduce these types of perturbations or these types of observations. So this is work in progress, and basically, you know, my point is that with models like these, one can do multiple types of studies, and this is really going towards multi-purpose applications, as long as you have enough biological complexity here. And by the way, in this case, And up to now, everything that I told you was probably more or less the same between the biophysical and point neuron version of the models. For applications like this, we are using biophysically detailed version. It's probably—
It's 10 minutes, Anton.
Thank you, Gaute. It's probably okay to use a point neuron here as well, although that's much more complicated. You need to make some additional assumptions. But you know, this is also a benefit of having multiple resolutions in models. So the last thing to mention is that all of this work has been supported by the tool development in our team, but also in the field. So we also share our tools for modeling freely. They are on GitHub. So there is this brain modeling toolkit, software that we use to interface with other existing amazing tools such as Neuron or Nest to run simulations. We also built the file format Sonata, working together in particular with the Blue Brain project and other collaborators. And this is something that allows one to share models, you know, between labs with the community. So yeah, you know, to conclude, we have this first version of the models of Mouse V1 that we developed. This is not the end for us. This is the beginning. This is the first step. And we really hope that we'll be able to create new versions of those models, increasing the accuracy and predictive power. Even just the thing that I mentioned, like the connectivity, new connectivity data sets from the Allen Institute that you will hear about later. These are amazing data sets that really beg to be included in models like that. And that's certainly something that we want to do in the near future. And then another thought here is that we are, I think, in a very special point now in the history of computational neuroscience. Just several years ago, if you wanted to do a modeling like this, it was much harder. There were some tools available, but in just last 5 years, there was really an explosion of new tools, of new software packages that different teams developed that permit one to run simulations like this and build models like this. So currently, tools are available. Models are available. So we develop these models. Other teams are developing other models. Many of them are being made publicly available. You'll hear about some of them later in this workshop. And for many applications, computational expense is also not that bad. So for example, for these point neuron models, one can run some of them on single CPUs, and that's quite affordable. So the conclusion here is that these multi-purpose models are becoming sort of ripe for applications. And we hope that together with theory and experiments, they can really make new contributions to the field and advance our knowledge of the brain. And we really encourage the field to use them more and embrace them and make predictions with them. So with that, I'll stop and thank our founder of the Allen Institute, Paul Allen, for his vision and encouragement and support, without which none of this would have been possible. And also all the amazing people working at the Allen Institute who made that work possible. And thank you for your attention.
So that's great, Anton. We have time for a few questions. There's one from like David Bloom here. Anton, can you elaborate on the subcellular dynamics captured by the biophysical model?
Okay, yes. So in the particular version of the model that we developed and made available, these are what we call perisomatic biophysically detailed models. So they have you know, the dendrites and the soma represented as this set of cables, but we keep all the active conductances at the soma. So in these models, there are no active conductances in the dendrites, and that really helps with computational complexity. But of course, it's a huge simplification also in comparison with what really happens in the real neurons. At the soma, we have about 10 different active conductances, you know, voltage-gated channels, calcium channels, potassium, sodium channels, a whole number of them, and they all are tuned to reproduce properties of different cells from our in vitro experiments.
Okay, and then there's a question from Audrey Sederberg. Anton, the initial connectivity you showed used in the model, drawn from work from Kuh, Hofer, Vogel, Peterson, and others, is similar but not quantitatively identical to the connectivity you showed from the Allen Institute. What difference does this quantitative difference in parameters make for the ability of your model to predict features of neural activity and perturbations to that activity?
Yeah, that's a great question. That's exactly what we would like to know. So, that's why the next version of the models need to be constructed, and we need to use this more recent data sets from the Allen Institute that obtained all this data systematically for all different cell types. I don't know at this point yet. There might be something really cool and amazing here, that when we swap the connectivity for this new one, then we will just all of a sudden the model will do better on certain metric. But it's also possible that it will not make such a big difference, right? And then there is a question, well, how this specific aspects of different types of connectivity, how they really matter. So yeah, that's a good question. We need to explore it. Stay tuned.
Here's one from Ben Huang. Are there plans to extend this approach to the rest of the cortex soon?
Well, I'd say no specific plans. This is something really, I'm interested in. Many other people at the Allen Institute and outside are interested in. There is some really cool work from the Blue Brain, actually, that also goes in this direction. So I think it will happen, but I don't have any specific plan or a timeline to say here yet.